Okay, so thank you everyone for coming. Sorry that it's been a bit chaotic, but hopefully everyone could find the slides. They should be accessible now. My name is Enny van Beest. I did my PhD in Amsterdam on visual processing, and currently I am at University College London, where I work as a senior research associate on multiple neuroscience problems. And I recently developed a software or a tool to track neurons or brain cells across many days or even months to years with high temporal resolution. And Cecilia thought it would be nice if I talked about this. Yeah, that's where I'm for today. And I'll try to keep it relatively short so there's some time for questions. The work was funded by EMBO, Wellcome Trust, and Marie Curie, which is part of the European Commission. And on slide 2, in the acknowledgements, hopefully you'll see that I did definitely not do this by myself. I specifically want to thank Cecile Tamura, who is also a postdoc in the lab. And we've been basically collaborating very closely on this project. And the two supervisors of the lab, Matteo Carandini and Kenneth Harris, have been instrumental in guiding us through this project, and a lot of other people from the lab. So here are their faces, and hopefully you recognize this work was a team effort. On slide 3, hopefully I'll show you why we even want to track neurons. So the picture is from a very talented PhD student in the lab who recently graduated, who trained mice to detect a stimulus. So the stimulus was on the lateral screen, and their task was to move it with a little Lego wheel to the center. So that basically with a closed loop. So if the mice turn the wheel to the right, the lateral stimulus would come in the center, and they would obtain a reward for this. And what we see if we record neurons in the basal ganglia from these mice, and the basal ganglia is typically associated with a lot of processes, but mainly motor initiation and maintenance, but also cognitive processes. We see that the responses to the central and the lateral stimuli are different for this population of neurons in this brain area. So we see at day 0, which is before mice were trained on this task, we see that the cells prefer to respond when the stimulus is in the center compared to when it's on the lateral screen. And then on day 2, we again recorded from this population of cells, and we saw that this didn't really differ, even though now the mice had an association with certain stimuli, because only when the stimulus was in the center, that's when they obtain a reward. On day 4, when the mice are really good at this task, we see that those two lines come very close together, which kind of suggests that they may have now coupled the action of moving the lateral stimulus to the center in obtaining a reward later. But of course, this is just the average response of a group of neurons in this area over multiple days. And from that, we would conclude perhaps that neurons in the basal ganglia increase activity to a rewarded stimulus first, and then to the stimulus associated with the action that leads to this reward. But we don't know at this point if that is true for all neurons. So on the next slide, we wanted to kind of illustrate why it would be interesting to track neurons at a single spike resolution, and why as a neuroscientist we find this problem very interesting. Because we want to understand what the neural code is doing when we make decisions, when we learn and forget, or even when we age. And to do this, we need to track the neural activity from individual cells over time. And if you can hopefully appreciate these cognitive processes, even though they take place in the order of seconds in the case of decision making, or hours if you talk about learning or memory, or even years if you're talking about aging, the neurons fire, so they communicate at the level of a few milliseconds. So that's quite a gap, going from a few milliseconds to seconds, minutes, or even years. So ideally, we want to track neurons at their firing rates, but for a long time. And one way that people have been tracking neurons is with imaging. So in this case, you bring a calcium indicator into these cells. So these are genetically modified animals where we can visualize the cells in certain brain regions. So the little light blobs that you see here are the cell bodies in specific areas. And you can see that you see the same field of view, the same cells in day 1 and day 38. But this technique, unfortunately, doesn't capture neural firing at the milliseconds that they actually fire. So it's always kind of a delayed average response, a bit similar to fMRI, perhaps, that you may have heard of in humans. So it's a very slow, relatively slow signal compared to the neurons firing. So on the next slide, I'm introducing NeuroPixels probes, which they are called NeuroPixels, hopefully, because you can see already that it kind of contains pixels. And these are recording sites where we can measure electrical activity on each of these black dots on this probe. And this gives us a high throughput way to record from many different neurons their voltage over time. And originally, these probes are always inserted in an acute fashion, which means you insert the probe temporarily for an hour or so. You record while mice are performing a certain task, for example, a decision-making task. You can analyze the activity, and then you get the probe out of the brain again. But of course, as I just said, many cognitive processes occur over the time of hours, days, weeks, or years. So ideally, we want to prolong this time. If you go to the next slide, you see that ideally we want to cover, make a tool that we can use this NeuroPixels probe, but use it for an extended period of time. And one way to do that is to implant it chronically. The first effort was actually an effort by people in this lab a few years back, where if you look at the brain slice on the left, you can see that this probe went into the brain via this kind of superficial area, which was a visual area in the cortex, then the hippocampus, and then the thalamus. It doesn't really matter which brain areas for now. And on the right side, you can see activity or proportion of spikes in each of these three regions as a function of days since implantation. And hopefully you can appreciate that the spiking activity is very stable over even 309 days, so almost a year of time. But this is when we chronically implanted this NeuroPixels thing, which means it's non-recoverable. So it means if the mice have not a very long lifespan. So if they die or you need to do histology on the brain, you lose the probe. And these probes are not super cheap. So we first, as a lab, try to design recoverable chronic implants. So that's what you see on the next slide. We designed it as a docking module and a payload module. And the payload module is going into the docking module. This is all 3D printed, and the probes are just kind of connected to the payload module. And the docking module is what's cemented on the skull of the mice. Now you can insert this as a whole, but when you're done with a certain mice, you can unscrew the payload module from the docking module. So you can recover the probes with the payload module, and the only thing you lose is the docking module. Now the docking module can be 3D printed for maybe £2 or $2. So that's relatively cheap, and you recover the probes themselves. So that's a very kind of nice way to do these chronic NeuroPixels recordings. There's many other examples of recoverable chronic NeuroPixels by now. So I just put an overview. There's an overview of all the different labs making different versions. A few things you have to think about is like the weight of the animal. So we work with mice, so we want it as light as possible so that it doesn't bother the mouse too much. And there's a few bigger implants which are only suitable for rats, for example, because they're a bit bigger. On the next slide, you can see that if you have these recordings in a recoverable chronic setup, you can record units for many days since implantation. So in purple, the two top lines, you see two individual probes. So from every probe, which has 1,000 contact points, we can record about 384 contact points at the same time. So these are the purple lines below. And if you add each of these together, you get one individual probe. So that's the difference in number. So from each probe, we can record about 1,000 units, slightly less, it's a log scale. And after a certain amount of days, typically you see a decline in the number of units you can record. This could be, for example, movement of the probe or kind of, what's it called? Like if you insert something in your skin, there could be some stuff going around
that thing you insert as an immune response. So we're not entirely sure yet what happens. So that was just one example animal. And if we do this for many animals, we see a similar effect. Now, this is just showing how many units can we record, but ideally you want to know exactly which units are you recording and which ones do you lose. So that's on the next slide, basically the question, how do we track these individual neurons in these populations over months? Before I go into that, I want to briefly explain what spike sorting is in general, because not everyone may know this. So if you imagine on this slide that says spike sorting and single recording, if you imagine a few neurons here in colored dots around the probe, and the probe is this great thing with the black recording sites, each of these neurons, the circles, gives a certain electrical activity that contributes to some of these recording sites. So the question of spike sorting is to figure out kind of what is the sources of the electrical activity on each of the channels. So this is a bit similar to the problem of having a lot of speakers and a lot of people in a room trying to figure out who said something if you record it on multiple microphones. So this is a similar problem which has been solved more or less. It's still not optimal, but it's solved more or less. And this is spike sorting. So what you're doing is you're getting all the spikes, which are the little vertical blips in the neural activity that is shown, and you attribute these spikes to specific neurons by basically matching how their waveforms look. And this will become clear later, I hope. So once you've done that, you can kind of color code these spikes by which neuron they belong to. So that's the spike sorting. If you now have multiple recordings where supposedly you have the same populations of neurons in these two recordings, initially there was a way to do this, which is now we pretend that these two recordings were recorded in the same day. So we're concatenating the two recordings. So this is on slide 13. And if you pretend they are recorded on the same day, and you concatenate the data together, you usually find spikes of neurons that you can find in both days. So this way you know which spikes on both days belong to which individual neuron. However, nowadays, I missed one step. On the next slide, you can see that if you apply this method to this original data that I showed you before, that this kind of works. And how we validate that it works is by looking at the individual neurons' responses to images. So this data was obtained in visual cortex, and visual cortex likes to respond to pictures. So we presented a whole lot of pictures and natural images they are called, for example, of mushrooms or spider or you name it. And we looked at the responses of individual neurons. So in the top row, we have neuron 70 and the bottom row, neuron 71. And we looked at the responses of these neurons on the different days. So the different colors are the different days. And hopefully you can see that the responses on different days are very similar when the same picture was presented from the same neuron. But they're very different if it's a different neuron. So this is kind of a fingerprint of what this neuron likes to respond to. And this way we can validate that the neurons we found across days are the same indeed, functionally as well. So the only issue with this method of concatenating the data together is that if you have a lot of recordings, so this is on the next slide. So with these new techniques we can record for multiple weeks to months from the same animal. So supposedly we're recording from the same populations of neurons for maybe hundreds of recordings. Hopefully you can imagine that adding all this data together and then spike sorting this is very inefficient to the level that it will not work. Your computer will just crash. So we needed another method. Okay, so the first recap is that semi-chronic neuropixel implants are ideal to capture neural activity across the brain from milliseconds to months and potentially years if we figure out some issues with probe durability. and that available methods for tracking the neurons are not scalable. And the alternative is using functional activity, but normally we want to answer questions about functional activity, so that's kind of circular statistics, so we kind of want to stay out of that. So on the next slide, I show you the pipeline we developed called Unit Match, which solves this problem in a more efficient way. And I'll go over each of these steps. Hopefully it's a little bit clear, but otherwise feel free to ask questions at the end. So on the next slide, you can see the pre-processing and what we're doing. So for each spikes recording we do the spike sorting individually. So now for recording N all the way to recording N1 to N, we have the data spike sorted. So we have a list of neurons for that recording and a list of their spike times. And we have that for all the recordings. And we then curate these neurons. So we make sure the neurons look nicely. So in case you wondered what the electrical waveform of a good unit should look like, the right top waveform plots show waveforms of good units. And hopefully you can see that they are very different from the kind of noisy unit waveforms that are more or less flat. And yeah, there's a lot of literature. So that's how we know when something is a good unit, when it's non-somatic, etc. What we then get for every neuron is a bunch of waveforms measured on the different recording channels, because remember we had 384 recording channels. So we can measure the signal from each neuron on each recording channel for the first half of a recording and the second half of a recording. So that's on slide, what slide is this? 18 on the right side of this form. And if you go to the next slide, you can see that you can do that for a lot of neurons, right? So we do this for every neuron. We have the first half of the recording, the second half of the recording, and we show all the waveforms on the different recording sites for the black neuron. We do the same for the closest neighbor in blue, and we do the same for another neuron in another recording session, the red neuron. Once we have all these waveforms from all these neurons, from all the recordings, so it's a lot of data, but it's a lot less already than the raw data, we're going to extract waveform parameters. So this is now the next slide, where we see extract waveform parameters. So I again show you the NeuroPixels probe. It's a schematic of it with recording sites. And next to it, you see each one neuron's waveform projected onto each of the recording sites. And hopefully you can already see that one of these waveforms is a lot larger than the others. So that's what we call the maximum site, because it's a site where we record the maximum signal for this specific neuron. If we now measure the distance of each of the recording sites to this maximum site, and we plot it against the amplitude measured on that recording channel, we see that you can fit this exponentially as an exponential decay. So you see that this amplitude declines with distance to this maximum channel. And where the signal is just 10% of the maximum response, that's the distance where we say, okay, this is until here, we can measure this neuron's activity. And beyond that, it's basically in the noise range. So for this specific neuron, we include only these channels that are colored black here. From these black colored recording sites, we compute the weighted average waveform. And from this, we also extract the weighted average amplitude. So there's the kind of strength of this waveform. On the next slide, you can then see that we also extract different features. So we have spatial decay so far, and we have the waveform and the amplitude. And we can also use the spatial information from this probe to have more information about this neuron's waveform. Okay, so this is a bit complicated, so I'll try to go slow. But if you can see now, I drew some kind of funny looking pyramid thingy on the left side, close to the probe. And this is kind of showing a neuron schematic. And the little round circle is what we would call an axon initial segment. And this is where typically a neuron's voltage starts being high, and after that the signal travels through this neuron. So because it moves over time, and we're talking here about 0.7 milliseconds, while this neuron is spiking, this signal travels within the neuron. And this, since we have so many recording sites that are very close together, we can measure a small movement of the signal and quantify it. So that's what we do with the centroid trajectory. For every time point in the spike, so from when the waveform starts going down and when it comes back up, that's where we measure where the signal is the strongest at that point in time, and that's the centroid trajectory. And we can compute the average position, and we can compute travel direction and travel distance in between each of the time points. Hopefully that was a little bit clear, but it is a bit complicated. But basically we have a spatiotemporal footprint of every neuron's waveform. So then to the next slide, what we're going to do is we're going to compare these extracted parameters for the different neurons that we have. So in A, what you can see is the closest neighbor within the same day. And you hopefully already see that the waveform is very different. The amplitude is a lot larger. And also this trajectory in B is very different. So we can compare how different or how similar are these two, are all these parameters that we extracted. So these are the similarity scores, and what they are is written below in C. And hopefully you can see that most of them are well below 1, except for C, which is the centroid position, because it's the closest neighbor. So it's very close by, so that gets a high score. But on average, the score of this neuron is relatively low. And if you do this for all the neurons by all the other neurons, so that's in G now. So how you should read this plot is every column and every row is an individual neuron. And we have all the neurons from day 1 and all the neurons from day 2. And we extracted their waveforms in the first half of that recording and the second half. And did we do this to kind of cross-validate what the same neuron within the same recording should look like in terms of similarity. So hopefully you see that there's a very high score on the diagonal. So this means that the neurons themselves look very much like themselves if we apply the similarity scores, which is a good sign. And that other neurons that are not the same within the same day do not look the same. So if you quantify this in H, the neurons on the diagonal, the same units, they have a very high total score, and the neighbors have a low total score. And it's quite obvious where you should put a threshold in order to find two neurons that are the same. Now, if we go to the next slide, you can apply the same trick in data that comes from different recording sessions. So now in red, we have a neuron recorded on a different day. And we already see that the waveform is very similar to the black neuron recorded on day 1. And also the spatial trajectory is very similar. And if we quantify this in similarity scores, we can see that the total score is very high. And hopefully you also see in G now that within the red boxes, so that's across days, we have a second diagonal showing up. So these are the neurons that are having really high total scores. And these are neurons that we're potentially interested in because they are actually very likely to be the same neuron recorded on different days. So what we do is we apply the threshold that we found in H, we apply it to the across day data in I, and then we say these ones on the right side are potential matches or potentially the same neuron, and everything else on the left is likely just a neighbor. But you probably can already see that this distribution is a little bit noisy, so just this is a difficult threshold to define. So what we're doing is we're going to do a small trick. So we're going to now use these putative matches, as we call them, and we go back to the original scores. So now we're on slide 25. And you see in J, distributions for each of the parameters that we have. So decay similarity, waveform similarity, etc. And you see the probability of a certain similarity score when the two neurons were higher than the threshold or lower than the threshold. And hopefully you can already see that these distributions are very different for the black and the gray group, so for higher than and lower than threshold. And these are what we call probability distributions. And because we make sure that they are not correlated, we can put them into a naive Bayes classifier. And together, this cleans up this matrix that you saw on the previous slide. So now, instead of a very kind of normalized, normal distribution of scores, we get almost a binary distribution of very high probability of two neurons being the same, or very low probability. There's almost nothing in the middle. And you can see this in L and M for within day and across day data. Of course, you find the neurons themselves back within the same day, but this is cross-validated, so that's a good sign. And you can find neurons across days that are having a very high probability of being the same. And these are now the ones that we think, okay, these are actually the same neurons. This is what we're interested in. So on the next slide, you can see that I try to kind of visualize this in graphically. If you have a bunch of neurons sitting on a probe, originally you don't know which of the gray neurons on day n match with the ones on day one. But now that we have this very high probability for some of them, we can match a large part of these neurons back to what they were on day one. Yeah, so that's is kind of what I just said on the recap. But how do we know whether these matched neurons are actually valid? Because we only use the waveforms, the spatial temporal waveform, and maybe we want to have a different method to validate that this is real because we don't really have ground truth data. So it's a bit hard to know if this method works. So what we did is we now looked at the functional activity of these neurons to validate that what we did with our algorithm works. So the first thing we do is we look at the distribution of interspiked intervals. So what this means is how long after a neuron spiked does it spike again. So that is a distribution with, it's a logarithmic X scale, so keep that in mind. And if you do this for one neuron in black for the first half and the second half of a recording, hopefully you see that these distributions are very similar. If you now do this for a different neuron in blue, these distributions are very different. And if we do it for the matched neuron across days, it's again very similar. So you can correlate these traces with each other, and then you get very high correlation when it's the same neuron, when it's a match across days, but not when it's the same unit. And these correlations are so different, these distributions, that you can use the correlation value to kind of assign or predict whether two neurons would be a match or not. And you can do that to the same level as predicting whether it was the same unit on the same day. So it's a very high kind of performance across days. And this AUC value, which you extract from this kind of classifier, remains very high. So for this example mouse on the right top corner of slide 28, you can see that all these dots lie quite high above the 0.5 line. And this is also a logarithmic scale, so we can up to 180 something days, we can find the same neurons, and we do that with quite high accuracy. And this was true for all the mice in our data set. On the next slide, we did the same approach for natural images, what I showed before. So you can again look at how do neurons respond to different images, what is their time course, what is the average response to these different images. And if you do that, you find again very similar responses for the same neuron, very dissimilar responses for different neurons. And you can use that to validate that the method works. Okay, so unit match performance was validated using a completely independent measure, which was functional stability. So how do we track the neurons across many recordings? Because now I showed you always pairs of recordings, but in reality, what you want to do is you have 100 recordings, and you want to know which neuron was present in which of these recordings. So that's what we call multi-day tracking, which is now slide 31. So on the left side now, you see this matrix that I showed before, but instead of for 2 recording sessions, I show it for 22 recording sessions. So you can see the big diagonal, which is all the same neurons on the same day. And you see off diagonals, which is basically the first off the diagonal is the difference between the difference of one recording. The second one is a difference of 2 recordings, etc. So the fact that you do see all these diagonals showing up shows you that with a large distance in between recordings, we can still find neurons with high matching probabilities. The way we now assign the groups overall across this entire data set is by looking at their probability. We sort them. So the highest probability gets a ranking of 1. So if you look on the right, that's the left neuron with the middle neuron top. This gets a ranking of 1 because the probability was 0.99. Then the second highest in the data set would be 0.8. So we rank it 2. Because all these connections are very high, we say this black neuron, or this was the same neuron, so we give it the same identity across the 3 recordings. And now we have a third highest ranking, which is 0.6. But unfortunately, on day 2, there was already a neuron assigned to the black group. And this blue neuron doesn't fit with the black neuron on the same day because the probability was 0.4. So we do not add it to the group, even though the probability between day 1 and day 2 was higher than 0.5. So this kind of is an extra check on top of the match probability across many days of recordings. On slide 32, you can see an example. So this is the same data. We have a reference day 68, where we have a bunch of neurons on the probe. So there's these colored dots. And if you go back in time, so day 59, day 24, etc., a lot of these neurons were found back that were present on day 68. But day 12, we happened to have a lot of noise in our recording. So we couldn't measure a lot of cells because the data was just not good. On day 118, we had a similar problem, like the data was just not so good, so you don't get a lot of neurons. The nice thing about this tracking algorithm is that it doesn't really care. So if a neuron disappears on one day because either it wasn't there, it wasn't active, or there was a lot of noise, you can find it back on the next recording so long as it comes back. And this is different from any other tracking algorithm where tracking was always done sequentially. So if you lose a neuron, you lose it forever. So that this tracking algorithm can handle neurons disappearing and reappearing. On slide 33, we're now showing for every neuron. So now we have all the neurons and they get a unique identity. And we show whether it was present in each recording. And if it was present, we give it a black color in this left matrix. So what you can see is that the first few recordings, there were a lot of neurons that were not tracked really well. So we often see this because initially we put in the probe in the animal. It may take a few days to settle in the brain. So there's a bit of a movement where the brain kind of resettles after a surgery. And we see this more often. But then once it's stable, we have quite a stable block of recordings where neurons can be found again day after day. So the chance of tracking a neuron is the top right plot on slide 33. If you have a group of neurons on day 0, or on reference day 0 in this case, and you look at delta days in the past or delta days in the future, the chance of finding the same neurons becomes less. And that's partly because, as I showed you earlier, the population of neurons declines over time. So you're going to find less and less neurons. However, since it's kind of almost a symmetrical distribution, so that also means you're going to find new neurons. So it's not just that neurons disappear, it's just new neurons come in, neurons that go out. And on average, we can track only 20% of the neurons, but we can track them for more than 128 days. So I think the leftmost tick is 200 days. So that's quite a long time. And you still find 20% of the neurons, so that's quite okay. Even within the same day, if you have an animal on different experiments, we only find 60% of the neurons back. And you might find that surprising, but maybe you've heard of this, which is slightly untrue, but maybe not entirely untrue. It's like you're not only using 10% of your brain. That's not really true, but there will be neurons more or less active in certain tasks, right? So if we put a mouse in a certain task, different cells will be more active, and only active neurons can be picked up with our electrode. So it's not super weird that we only find 60% of neurons if we record the mouse on the same day in a different task. So 20% in 200 days, we were quite happy with that. We validated this again with these AUC scores that I talked about before. It's always above chance for both the interspike interval distributions and the visual responses, chance being 0.5. And the bottom plot just shows the number of data sets that went into this, because we didn't record all the mice for 200 days, unfortunately. Okay, so we can use the unit match to track neurons across many recordings, even when they temporarily disappear. So that's great, but what can we do with it? So on slide 35, I'm going back to this original experiment that I talked about. And now, hopefully you can see that more plots were added. So in C, we have 3 example neurons now, where we track the waveform. In D is their interspike interval distributions. So this hopefully convinces you that this indeed are 3 units or neurons that are the same across 3 recordings. And hopefully you can see that individual neurons can do very different things than the average population that was shown in B. So for example, the neuron unit 72 in the top row in E has a response only to the lateral stimulus and never really has a big response to the central stimulus. The same is true for unit 89 or neuron 89, and only 158 in these 3 examples has a big response to the central stimulus. And you see that the average response in B was completely driven almost by this unit 158. So this kind of shows the importance of finding the same cells so that you can conclude a very different thing than what you would conclude from looking at just the population average. So yeah, unit match can be used to track single neurons over learning. And what I'm going to do next with this is to, this is now slide 36, to have mice. Sorry, so I'm having mice doing a virtual reality task. So they're on a ball and they have to run through an environment. And what I want to ask is, do we have units that care about where we are in space? And are these the same units? If I know the same neurons, do they care in this linear environment versus when I put them in a freely moving 2D environment? Or is it maybe very different neurons? So this is what I'm doing now. So I'm using these chronic implants and can use the mice in different tasks and ask these kind of questions. So these are questions that we couldn't answer before. So on the next slide, you see 2 example neurons that I have identified in these 2 environments. So the first neuron on the left really likes the location B a lot. So every time it's close to location B, it fires a lot. So that's seen as a black spikes per second in the top graph, and the average is below it. And on the right side, we have another neuron that also kind of likes B, but it's only firing there and it's very silent otherwise. These same neurons have a specific place in this freely moving environment where they also like to fire. The top one has a very broad spiking field, so as long as the mouse is kind of on the left side of this freely moving environment, it will be active. Whereas this other neuron has a very localized firing field, so only in the left bottom corner and this kind of middle spot and maybe one spot on the right, it's firing, but otherwise it's silent. So that's what I'm kind of working on right now. So this is just a kind of a teaser. And there's many more applications of unit match. So we're looking, like there's many people in the field now using this technique to answer a lot of different questions. So we're really excited to see what comes out. Yeah, and on the last page, last slide, I just put some links, some QR codes. The code is available in MATLAB, Python, and it's also compatible with most used data analytics tools in the field. We have some graphical interfaces to look at the matches and see if they make any sense. And we can also use it to do within the controls of quality control, basically. If people want to try it out, there are some tutorials online with some example data. And I also would like to thank Cecile Tamura again, because as I said, we did this work together. And yeah, this was very important. His work was very important in this. So please, yeah, keep that in mind. I think that's it for me. So if you have any questions, I'm happy to answer.
Thank you so much, Annie. And I apologize for having been late to you and to everyone, every member of our audience. So I thought that, I could wing it by trying to start the room from the outside. I had to go to the hospital because you see, I was having some flu about a week ago. And there was a recurrence of the flu, so I got a little bit of panic and had to go at a time that I have not scheduled. So I'm very glad that Bob and Darcel had opened the event. And thank you very much, Annie, for the great presentation. We'd like to open the floor for the audience if they have questions for Annie. Yes, so I have a question. So this study, could this method be extended to study more complex behaviors or conditions such as memory formation or neurodegenerative diseases? Maybe you have answered that, but I was late. So, anyway, I was having this question in my mind.
Yeah, thank you, and no problem for being late. I hope you're okay, and take it easy. It worked out, so no worries. Yeah, so I mean, it could work to study these things, but we're always looking at spiking activity, right? So in terms of, we can look at, do functional activity of these neurons change as a function of disease or memory. However, some of the things that will happen in these, in memory or in diseases, is that neurons might completely disappear, for example, or they might change in a different way. So for some of these things, you may need different techniques, more kind of biochemical techniques, or looking at the DNA expression, that kind of stuff. So it's definitely, we can definitely use it. The issue is when a neuron is silent, we don't know if it's just not spiking and we don't pick it up, or if the neuron is completely gone because it was sick. So for this specifically, you need different techniques. But we can definitely say it's spiking differently, or it's spiking and not anymore. That's something we can uh, say with this technique, if that makes sense.
Yeah, thank you. Annie, you had mentioned a while ago that is this technique of yours open to the public?
Yes, so we have code on GitHub, which is under, it's licensed for free use except commercial use. For commercial use, you can still use it, but then you need to contact UCL business, because, yeah, just for kind of making sure everything's fine. But in theory, it's open source, so yeah, it's available online.
Yeah, so I'd like to make an applause for open source because I think you are so generous with the results of your study. It will lead to more discoveries in the future, and this is great for humanity to understand the human brain further. We could see that there's already a big difference of what has been studied, even for the past maybe 8 years. There's a significant difference in neuroscience because of new technology and studies that you did. So yeah, I'd like to give an applause to your group and gratitude also because this will help us all. So
Yeah, no, that's a main reason for me to be in this group. I think my two bosses, they really make a point of helping the community and being open source, so that everyone in the community can benefit, and we as a field advance the understanding of the brain through many different tasks or diseases or cognition. So yeah, it's for us very important that at least scientists can use it and people that are interested can use it. Yeah, so I'm happy to contribute a little bit to that.
yeah, thank you again, Emmy. Kevin, Kevin is here on stage, and I'd like to pass the mic to him if he has some questions.
Oh, hi, Enny, and yeah, hopefully you're fine, Cecile. That's very concerning, of course. Oh man, right before
At
Christmas.
least it's COVID, so I already tested. So yeah.
Oh, it was not COVID.
No.
Oh,
You
oh, man.
Anyway, I pass the mic to you.
Oh, no. Oh yeah. Well, hopefully, I said right before Christmas, but yeah. Oh man, it makes me think of CRISPR now, but I don't know if that has to do with this. But okay, yeah, yes, maybe some. I was thinking of unit match. Um, the, do you know if how would how do you think that may address like some existing challenges in neural data analysis? And where are some advantages of using this compared to traditional methods? Just some things I thought of first.
Yeah, thank you for your question. I think the main reason I developed this is because initially we had a lot of data from mice with these chronic implants, and we could look at population statistics. So that's kind of why I put the example of Julie there, who had the population data, and we could answer something about the population, so what a group of neurons does. But what we didn't know is what these individual neurons, how they develop their activity over learning, for example. So this is a technique that for the first time can help us solve this in a large data set of tens to hundreds of recordings, which was just not that it wasn't there like 2 years ago. So that definitely opens up the field for many questions like related to learning, related to memory, maybe related to aging. So yeah, I think a lot of data that has been acquired recently can now be reanalyzed with different questions in mind about what individual neurons do and how they act together as a population, rather than just looking at an average population, if that makes sense.
Oh yeah, and then I was thinking of plans for integrating Unimatch with other tools or platforms, and maybe how it might be maintained and supported in the future. Some other questions I thought of, general.
Yes, yeah. So for us, it was important that as many people could use it as possible. And originally, because I'm more of a MATLAB coder, because I guess I'm from a certain age, I learned to code in MATLAB, and this is where I wrote it in initially. But we are aware that many younger scientists prefer Python because it's also open source. So we have translated UnitMatch to a Python version, and we also made it compatible with Spike Interface. And Spike Interface is kind of a large platform for analysis of electrophysiology data, and we're in close contact with the developers of Spike Interface. And they have a large group of people whose main job it is to maintain this. So we're hoping to kind of lift on that a little bit, because yeah, indeed, it's not my main job to maintain software, but I'm happy to do it for UnitMatch. But at some point, if I move somewhere or do another job, it's great that it's integrated in a platform where people are dedicated to do this. So that's why we chose to integrate it with Spike Interface.
Oh, okay, and then as far as the study, the other thing I thought of and looked up is were there any did you address any limitations in calcium imaging? Is what I could see.
Yeah, so like calcium imaging is a tool where you, for a longer time already, could kind of follow neurons over a long time. But as I said in the introduction, I think the issue with calcium imaging, well, there's two things that make it non-ideal for tracking individual neurons. The first is neurons fire at a few milliseconds. Like one spike is less than a millisecond actually. Whereas calcium imaging has a slower time scale. At best, it's like tens of milliseconds. So you cannot capture neurons at their normal communication kind of temporal frequency, right? The other issue is that for imaging, you usually have to stick to the top layer, so the outside layers of the brain, because that's all you can image optically. If you want to go to a deeper region, like for example the hippocampus or the basal ganglia, you can implant a lens, but these things are about 300 micron at least in diameter. meaning that you have to remove quite a large part of the outer layer of the brain in order to see the neurons in the inside layers. With neuropixels, it's a very small electrical probe. So sure, you will still pierce the brain, but the damage you do is a lot less than implanting this glass lens into the brain. So you can access way more area of the brain with electrophysiology. So there's kind of the two aspects why I think, uh, yeah, electrophysiology and then tracking neurons with that would be, for most cases, more interesting.
Yeah, I'd like to ask Bob. I think he sent me a message. He mentioned that Annie has a tight time frame. Bob?
Yes, of course. Yeah, sorry, I need to go back to doing experiments later today. So yeah, if there's a few final questions, I'm happy to take them, and otherwise, yeah, I should at some point get back to work, if that's all right.
Yeah, I just have maybe one or two more questions. So I'm just very curious if techniques from other fields, such as computer vision or machine learning, enhance the neural tracking further. And what do you think? What synergies might emerge if we do that? If you thought about this possibility, and how might this neuron tracking method inform our understanding of artificial neural networks and their potential for long-term adaptation? That's all. Thank you.
Yeah, thank you. That's very interesting questions. I think the first question I can probably answer. The second is very hand wavy. The second answer will be very hand wavy. But the first answer is that definitely using artificial intelligence and deep learning might improve tracking of neurons. So we're actually looking into that at the moment. There's two aspects, or well, there's one main aspect that um, why it may still be nice to use this specific method, and that is that what we're doing is we're looking at engineered parameters, right? So I basically designed these parameters based on what I know about spike. Um, so it's interpretable. So if we have neurons that are matched, we can go back to these parameters and say, this is why it's a match, this is why it's not a match, and I understand why. If you use deep learning, it's gonna use the same data, but we don't exactly know what it's extracting. So this is the downside, I think, of deep learning. But of course, if it's a lot better in accuracy and we can validate it more accurately, then that will be the future. So we are looking into this at the moment. To your second question, I'm not sure if we can use it for making better artificial intelligence, to be honest, because there, I think you will always know if you have units. I think you will always kind of know the unit's identity because they are designed or they're created, right? So I don't think you'll need it in artificial intelligence, but
Yeah,
we can use artificial intelligence, but we will not help it improve, I think. Yeah.
thank you. So there's another person who came on stage just a while ago. Do you
Okay.
still have
Yeah.
time to answer his question, Annie?
Yeah. Yeah. Go ahead. Yeah.
Yeah, there is Prem who's on stage. We give you the opportunity. Thank you, Prem. I found her while searching wildly on my own, so I'm sure that you will find her. Okay. So
Thank you.
anyway, I'd like to thank our guest speaker, Annie, for the very detailed presentation here. And, quantum photonics is quite strong on neuroscience. This is one of our extensively discussed topics. And yeah, we are very proud to have you here. And maybe in a few days, we will have your event here, your presentation in our YouTube channel. So anyway, I will message you with that if it's already out. It will have a sync of your slides with whatever you're saying. And thank you. And I'd like to pass the mic to you if you have some last message for the audience.
Yeah, thank you so much for inviting me. It was very exciting. And if anyone has more questions or ideas, yeah, do get in touch, and I'm happy to answer them offline as well. Or if you have comments or anything, feel free to get in touch. And thanks so much. It sounds like a great community and a great series of lectures. So well done.
Yeah, thank you so much. So, yeah, I'm very proud of my community here indeed. To date, so far when I started curating, we have interviewed already more than 600 researchers, serious researchers like you, including three Nobel Prize winners. And these researchers come from eminent institutions, and we are known for scholarly presentations. So we are very proud to add you uh, among our guests. And I'd like to pass the mic to Bob if you have some last comment, Bob.
I just wanted to say thank you for really interesting slides.
Yeah, thank you, Bob.
Thank you
Also,
so much.
also please note we have an upcoming event. I think in the morning again. So yeah, it's also very interesting. And now I'd like to pass the mic to Darcel to give us the guide to do the countdown to close the room.
Thank you, Cecile, and thank you, Enny, for a wonderful room. Um, to close the room, we're going to start our countdown from lucky number 6. Is everybody ready?
Eddie.
Ready?
6,
6,
6.
Prim 5. No Prim?
Sorry,
5,
5.
There we
Sorry.
go. Kevin
I don't know
4.
how to say something. Sorry, man.
Quattro, yeah.
Bob 3.
3 for me.
Cecile Dos Equis.
2X.
Good doctor, your numero uno. Please call out our last number, please.
Who won?
Thank you so much, Annie. We hope to see you back here in Quantum Photonics. And thank you, everyone. I'll see you in the next rooms.
Thanks.